Welcome to this week's Short Clip Friday podcast. Each Friday, I'll be posting 10 to 15 minute segments from previous podcasts. And the point here is just to bring you some of the highest yield topics from each podcast in order to kind of reinforce those topics. This clip is going to cover enzyme types and is from the 6th podcast that I did on enzyme kinetics, inhibition, and categorization. Enjoy. All right, so there are 6 subtypes of enzymes that you will need to know for the MCAT. The first are called oxidoreductases, and these catalyze oxidation-reduction reactions. That's pretty easy to remember. Now, the second type of enzyme you need to know is called a transferase, and this also does something that it sounds like it would do. It catalyzes the transfer of functional groups between different molecules. And let's see, number 3, the third type of enzyme you need to know is called a hydrolase. And so a hydrolase cleaves bonds using water. And then the fourth type is called a lyase, and that catalyzes the breaking of various chemical bonds by means other than hydrolysis or without water. So a lyase, remember that it just breaks shit apart without water. And number 5 is called an isomerase, and so that catalyzes the transfer of functional groups within a molecule, so it makes isomers. And last, you have an enzyme called, this is number 6, by the way. This is the last one you need to know. This is called a ligase, and so that joins 2 molecules together to make an even bigger molecule. And all these enzymes are relatively important to know for the MCAT. You might get a question where it shows you some substrate and some product, and it says, what kind of enzyme was able to, catalyze this reaction, or, and it's pretty easy though. I mean, you'll see one molecule and another molecule, 2 substrates, and then you make one product. Oh, what is that? It's probably a freaking ligase. Right, so that's easy. What is more important, in my opinion, are to know some common biological enzymes. And so I'm going to talk about dehydrogenase, synthetase, synthase, and phosphorylase, kinase, phosphatase, and I'm going to give a few examples of what these do. All right, so the first biological enzyme is called a dehydrogenase, and so a dehydrogenase removes a hydride, which is just an H group, hydrogen. It removes a hydride from a molecule and transfers it to an electron carrier. So, and these are usually NAD plus or FADH. So, dehydrogenase takes the NAD or the FADH and makes that into NADH, which is an electron carrier. And if you're thinking, wow, that sounds like an oxidoreductase, then you are very smart and you are also very correct. A dehydrogenase falls under the larger umbrella classification of oxidoreductase. The next biological molecule is called, or biological enzyme is called a synthetase. Synthetase. It's got an E in the middle of it, which differentiates it from synthase, which is the next one. Anyways, a synthetase links 2 molecules together using the energy from ATP. And it is an enzyme that plays an essential role in the metabolism of nitrogen. And it does this by catalyzing the condensation of glutamate and ammonia to form glutamine. And again, it uses the energy of ATP. And so a synthase, on the other hand, links two molecules together without the use of ATP. And so a good example of this is actually ATP synthase. And so this is an enzyme that creates ATP. So again, you're not using ATP to create ATP. So that enzyme creates ATP, and this ATP is then used and a lot of different other biological processes. And so this ATP synthase essentially takes adenosine diphosphate, or ADP, and an inorganic phosphate, which is PI, and combines them to make ATP. And again, this is the transmembrane protein that is the last complex in the electron transport chain. Moving on, the next biological enzyme you're going to want to know is a phosphorylase. And so phosphorylases catalyze the transfer of a phosphate group to a molecule from an inorganic phosphate. So an example of this is glycogen phosphorylase. So glycogen phosphorylase catalyzes the rate-limiting step in glycogenolysis in animals. And so it does this in animals by releasing glucose-1-phosphate from the terminal alpha-1,4 glycosidic linkage. The next biological enzyme you're going to want to know is called a kinase, and this transfers a phosphate group to a substrate from ATP. So the most important difference between a kinase and a phosphorylase is that a phosphorylase transfers this phosphate group, transfers a phosphate group to a molecule from an inorganic phosphate. Kinase, on the other hand, transfers that phosphate group to a molecule from ATP. And so a good example of a kinase is protein kinase A, and so this phosphorylates a bunch of different proteins and basically makes these proteins active. So like enzymes, protein kinase A might phosphorylate an enzyme, and then boom, that enzyme is activated. All right, moving on, we have another very important biological enzyme called a phosphatase. And a phosphatase uses water to cleave a phosphate group off a substrate. And this goes the opposite direction of the kinase and the, the phosphorylase. So a phosphatase uses water to cleave a phosphate group off a substrate. An example of this is protein phosphatases. And so these dephosphorylates amino acid residues and deactivates proteins. So these act basically directly against a protein kinase. So that's just kind of a brief summary of the biological enzymes you're going to need to know. And, those are important for a lot of these biochem questions. Off the top of my head, I think it's going to show some different pathways. You'll see I think there's a lot of kinases in particular, but those are all important to know. So the last thing that I want to talk about in this whole podcast is restriction enzymes. So restriction enzymes are DNA cutting enzymes, and each enzyme recognizes one or a few target sequences and cuts DNA at or near these sequences. And cutting typically results in single-stranded overhangs, which are called sticky, and or sticky ends. They kind of have like an L shape. They're called sticky because they can snap back together with their complementary base that they were cut with, or other strands that are complementary to their sequences. And most of these restriction enzymes cut at palindromic sequences, and so these are sequences that are read forwards and backwards at the same as the same thing. So essentially, if you take one strand, you flip it around, and then it should match that top strand. And so I'm sure you've seen the tattoos that are palindromic. For instance, one of my buddies has a palindromic tattoo on his forearm, and so it says his last name And, if you're looking straight at him and he has his forearm, in a normal human position, it reads his last name. And if he was to flip his forearm around and have his hand, pointing outside away from his body, it says the exact same thing. It looks the exact same way. So that's a palindrome. And go Google that if it still is not making sense to you. Restriction enzymes have a lot of uses in biochemistry. You can build plasmids. You can insert genes into these plasmids. And then these plasmids can be carried by bacteria. You can produce proteins, etc. And another interesting application of restriction enzymes are that they detect mutations. So let's just say that you have a genome and it contains a bunch of recognition sites for some particular restriction enzyme. Well, if some kind of mutation happens and now this recognition site is mutated, the DNA is not going to be cut at this site. And so what happens is you run one type of restriction enzyme, and let's say you get 4 different fragments of your DNA genome, and if you get a mutation, one of those sites is mutated. Now you don't cut the DNA at that point, and let's say now you have 2 fragments, and so you know that there was some kind of mutation in that DNA. And usually you'll see this as bands on a gel. So, this might be talked about in a different podcast, but when you run DNA on a gel, it'll separate it by size. So you have a mutation, you're going to have 2 bigger pieces of DNA because you don't cut it instead of 4. It's pretty easy to see on a gel.